Imagine for a second, that you are in your 20s or 30s and you have just survived this grueling, absolutely terrifying battle with cancer.
Yeah, you've been through hell and back.
Exactly. You made it through the chemotherapy, you beat the odds, and you are finally ready to just, start living your life again. But then you were hit with this devastating side effect.
Yes, secondary fallout.
Yeah, because the very chemicals that saved your life have just completely decimated your ovaries. So you are suddenly thrust into chemically induced menopause. I mean, your fertility is stripped away, your hormonal health is fundamentally altered, and you're dealing with systemic issues like bone density loss and cardiovascular risks decades before you ever should be.
It's an incredibly cruel paradox, honestly.
Yeah.
I mean, you win the fight for your life, but you lose a core pillar of your biological vitality in the process.
Yeah, it's heartbreaking.
It really is. And in the clinical world, we're talking about premature ovarian insufficiency, or POI. And historically, reproductive medicine just hasn't had much of an answer for this. It's basically been a wall that patients and doctors just hit.
But today, we might actually be looking at a way to break through that wall. Welcome to our deep dive. We are pulling apart a massive, fascinating stack of medical literature today to see if a cutting-edge treatment, specifically exosome therapy, can actually serve as a primary non-hormonal regenerative option for young women experiencing this medically induced menopause.
Which is a huge paradigm shift.
Yeah.
And to do this, we are synthesizing everything from the foundational best practices of the American Society for Reproductive Medicine, the ASRM, and the European Society of Human Reproduction and Embryology.
Right, the major heavyweights in the field.
Exactly. And we're pairing that with the absolute frontiers of stem cell biology. We also have data on active human clinical trials that are recruiting right now, and we are going deep into the strict FDA regulatory frameworks outlined by Exovera Sciences.
Okay, let's unpack this, because to really understand the promise of regeneration here, I feel like we first need to look at the sheer scale of the damage that chemotherapy inflicts on that ovarian microenvironment.
Yeah, we have to look at the actual mechanics of the toxicity, because drugs like cyclophosphamide and busulfan, they are incredibly aggressive.
Right, because they have to be. They're fighting cancer.
Exactly. They are designed to seek out and kill rapidly dividing cells by, cross-linking their DNA or just disrupting their basic functions. But in the process of doing that, they cause massive oxidative stress in the ovaries.
So it's like a scorched earth tactic.
It really is. Specifically, they attack the granulosa cells and just force them into apoptosis, which is programmed cell death.
And without those granulosa cells, the entire systemic loop just collapses, right? Because they are the essential support network for the eggs, the follicles.
Up to the nurse cells.
Yeah, so when they die off en masse, your brain senses that the ovaries are failing, so it starts screaming at them to work by pumping out huge amounts of follicle-stimulating hormone, or FSH.
Which gives you a really stark clinical picture. You see sky-high FSH, while estradiol and anti-Mullerian hormone, AMH, they just absolutely plummet.
The feedback loop is completely broken at that point.
Completely. I mean, AMH is the standard marker for ovarian reserve. So when you see it flatline alongside that high FSH, the biological machinery has essentially stopped.
And when we look at the ASRM and ESHRE best practices from our sources, the traditional response to this is entirely compensatory, isn't it?
Yeah, entirely. They offer hormone replacement therapy, HRT, and perhaps in vitro fertilization if there's any reserve left.
Which, I mean, reading through those protocols, it sounds like traditional HRT is just painting over rust.
That's a really good way to put it.
Right, because it manages the peripheral symptoms, it mitigates the bone density loss, it provides some cardiovascular protection. It makes the house look okay on the outside, but the internal structure is still actively failing.
Right, you aren't fixing the ovary itself.
Exactly. And IVF in this context seems like, I don't know, trying to harvest apples from a dying tree. Like, you can pump the patient full of stimulation drugs, but if those granulosa cells are gone and the reserve is depleted by chemo, there's simply nothing to retrieve.
And the ASRM guidelines clearly show the futility of those traditional stimulation protocols when the ovarian reserve is that critically low. You just cannot force a chemically sterilized apoptotic ovary to produce healthy follicles just by flooding it with exogenous hormones.
The tissue is just too degraded to respond.
Exactly.
Let me ask you this, because this is the dogma we always hear. Has the damage from chemotherapy historically been considered truly permanent? Because we're always taught that a woman is born with all the follicles she will ever have, and once that tissue is destroyed, it's game over. It's irreversible.
Yeah, that has been the foundational assumption of reproductive endocrinology for decades. You manage the systemic fallout, but you do not fix the organ.
Wow.
But, when standard therapies cannot heal the roots of the tree, as you said, the focus shifts to regenerative medicine. And for a long time, the holy grail there was mesenchymal stem cells.
Right. Live stem cell therapy.
But what the frontier of the literature is showing us now is that the real breakthrough isn't actually injecting the live stem cells into the ovary at all. It is isolating what those cells secrete.
Okay, so if live stem cells are, say, too large or perhaps too fragile to survive in a toxic chemoblasted ovary, the workaround is to harvest their secretions. We are talking about exosomes.
Yes, exosomes.
I am seeing this term everywhere in the regenerative space right now, but we really need to look at the physics of what these actually are.
Let's look at their structure. Exosomes are microscopic extracellular vesicles, and I mean incredibly tiny. We're talking ranging from 30 to 150 nanometers in size.
It's practically invisible even to most microscopes.
Right. They're naturally secreted by mesenchymal stem cells, typically sourced from human umbilical cord tissue or adipose tissue. And the funny thing is, decades ago, scientists assumed these vesicles were basically just cellular exhaust.
Like just taking out the trap.
Exactly, just a mechanism for throwing out waste.
But the literature we're looking at shows they are the exact opposite of waste. They are this advanced biological communication network. The sources specify that exosomes are entirely acellular, meaning they do not contain live cells, they don't have nuclei or DNA that can replicate.
Right, they're just carriers.
Yeah, instead they are packed with active biological cargo. We are looking at messenger RNA, various microRNAs. The sources specifically highlight miR-146A5P and miR-21-5P, along with specialized proteins and anti-inflammatory cytokines.
And that lack of a live cell is the critical advantage here. Because when you transplant live stem cells, you introduce immense biological variables into the patient.
Like what? Rejection.
Yeah, there is a persistent risk of immune rejection. There's the risk that the stem cells could maldifferentiate or even generate tumors in a highly inflamed environment. Because exosomes are acellular, they present a minimally invasive approach with a vastly superior safety profile. And they have incredible biological stability.
And tell me if this analogy tracks. If the stem cell is the actual massive manufacturing plant that makes everything the body needs, the exosome is like a USB flash drive the factory sends out.
I like that.
It's just this tiny lipid envelope carrying the highly specific software update that the damaged ovary needs to reboot its systems.
That is exactly what it is.
But wait, that raises a logistical question for me. If the stem cells are the actual factories generating these regenerative signals, wouldn't we want the factory on site? Like, why settle for just the flash drive when you could have the whole manufacturing plant localized right there in the tissue?
It seems counterintuitive, I know, but you have to consider the microenvironment of a chemotherapy-damaged ovary. It is highly toxic, flooded with reactive oxygen species, and profoundly inflamed. Live stem cells simply struggle to survive in that hostile terrain.
Oh, so they just die off before they can even do the work.
Furthermore, because stem cells are physically large, when they are administered intravenously, a huge percentage of them get trapped in the pulmonary capillary beds.
Wait, they get stuck in the lungs?
They literally get stuck in the lungs. Or they just get flagged and cleared by the immune system before they ever even reach the pelvic cavity.
Ah. And the exosomes bypass all of that because of their scale and their lipid bilayer.
They slip right through. Because they are nanometer-sized, they easily penetrate tissue barriers. And crucially, they do not carry the major histocompatibility complex markers. Those are the cell surface antigens that trigger an immune response.
So the body doesn't even see them as foreign invaders.
Exactly. Their lipid bilayer allows them to travel cleanly through the bloodstream, dock directly onto those damaged granulosa cells in the ovary, and just fuse with them, depositing that regenerative payload right into the cytoplasm.
Here's where it gets really interesting. Let's trace exactly what happens when that payload drops, because the animal models in these studies are just staggering to read. How does a nanometer-sized vesicle reprogram a chemically destroyed ovary back into a functioning organ?
The researchers set up a highly controlled model. They took mice and rats and administered the exact same chemotherapy regimens, cyclophosphamide and busulfan, to induce a precise model of POI.
So they basically forced these animals into early menopause.
Yes. The animals' estrous cycles ceased completely. The granulosa cells underwent mass apoptosis. The hormonal profiles mirrored human menopause perfectly.
Okay.
Then they administered human umbilical cord mesenchymal stem cell exosomes, either via an intravenous tail injection or via a direct intraovarian injection.
And the mechanism of action detailed here goes deep into the cellular signaling cascades. The literature points heavily to two things: the PI3K pathway and the hippo signaling pathway. Let's break down the PI3KKT side first. You mentioned the microRNA cargo earlier, specifically miR-21-5P. What is its mechanistic target inside that damaged ovary?
So inside the ovarian tissue, there is a regulatory gene and protein called PTN. You can think of PCON as a heavy, restrictive brake pedal.
Okay, a brake pedal.
Its primary function is to suppress cell proliferation and keep primordial follicles dormant, basically preventing them from growing uncontrollably. But in a post-chemotherapy ovary, the environment is so damaged that this regulatory system essentially locks up.
So the brake gets stuck.
Exactly. The tissue freezes in a state of decay.
Yeah.
But the exosomes deliver that miR-21-5P, which directly targets and suppresses PTN.
It physically removes the brake pedal.
It removes the brake, which subsequently activates that PI3K-AKT pathway.
Wow.
And this is a massive survival cascade. It sends an overriding signal to the surviving granulosa cells to halt apoptosis, begin dividing again, and wake up those dormant follicles.
Wow.
It completely overrides the cell death trajectory initiated by the chemotherapy.
That is wild. And what about the hippo signaling pathway? Because the sources highlight miR-146A5P as another vital piece of cargo in that USB drive.
The hippo pathway is fascinating. Under normal conditions, the hippo pathway acts as an organ-size control mechanism. It restricts tissue growth and promotes apoptosis to ensure our organs don't just keep growing indefinitely.
Which is obviously a good thing normally.
Right. But when an organ is severely damaged and needs to rebuild rapidly, you need to temporarily turn down that hippo signaling. The miR-146A5P delivered by the exosomes helps modulate this pathway while simultaneously acting as a potent anti-inflammatory agent.
Oh, so it clears out the collateral damage.
Exactly. It quenches the oxidative fire left behind by the chemotherapy, creating a quiet, permissive environment where the tissue can actually use those PI3KKT signals to physically rebuild.
I mean, the results of this dual pathway manipulation are just difficult to overstate. Reading the data after the exosome injections, these chemically sterilized animals regained regular estrus cycles.
Which is huge on its own.
It is. The systemic feedback loop was restored, the FSH dropped back down to baseline, and the estradiol and AMH recovered. They physically reversed the follicular apoptosis.
But, and I'm looking at the endpoints of these studies here, this wasn't just a cosmetic improvement of their endocrine profiles on a blood test.
No, the endocrine recovery was just the precursor. The researchers paired the treated, formerly sterile females with males. And the mice that received the exosome therapies successfully conceived and delivered healthy, live offspring.
Wait, really? Chemically sterilized animals had babies again?
Yes. Their fundamental reproductive capacity was fully restored. Moving from a completely sterile chemoblasted state to a live birth just by delivering a highly specific biological software update via a lipid vesicle, that is, it is profound. I also noticed in the clinical practice sources that some researchers are combining these exosome treatments with platelet-rich plasma, or PRP. What is the biological rationale for combining those two?
It really comes down to optimizing that therapeutic microenvironment. PRP is autologous, meaning it is derived from the patient's own blood, and it is densely packed with natural growth factors and cytokines.
Right. PRP is used everywhere in orthopedics and aesthetics now.
Exactly. So if the exosomes are providing the highly specific epigenetic instructions, the precise blueprints for cellular repair via those microRNAs, the PRP is providing the raw materials and a biological scaffold to build on.
Ah, I see.
By marrying the targeted instructions of the exosome with the broad-spectrum growth factors of the PRP, you just amplify the regenerative signaling within that damaged ovarian tissue.
Okay, but if this combination therapy can effectively reverse POI and restore fertility in mammalian models so completely, the immediate question for anyone listening is obvious.
Why isn't it everywhere?
Exactly. Why isn't this the standard of care today? If a young woman finishes her chemotherapy tomorrow, why can't her oncologist order an exosome IV or a targeted intra-ovarian injection? And I guess that brings us face to face with an incredibly complex and strict regulatory landscape.
It does. The basic science is advancing at an exponential rate, but regulatory bodies, specifically the FDA in the U.S., they operate on a rigid framework of safety and proven efficacy.
Which takes time.
Lots of time. To really grasp why this isn't in every clinic tomorrow, We have to look closely at the regulatory documentation provided by Exovera Sciences, which breaks down the precise legal classifications of these biomaterials.
Yeah, the Exovera regulatory reference gets deep into the mechanics of FDA oversight. It draws a hard line between a 361 HCTP classification and a 351 biologic. Let's translate that for the listener. Starting with that 361 pathway, what does that actually mean?
So in the United States, human cells, tissues, and cellular and tissue-based products are regulated under the Public Health Service Act. The 361 pathway is reserved for tissues that are minimally manipulated,
Okay.
and they are used for homologous use, meaning they perform the exact same basic function in the recipient as they did in the donor. A classic example is a simple bone graft or a blood transfusion. Because the tissue is essentially unchanged, it does not require clinical trials or pre-market approval. A lab simply registers the product and lists it.
And looking at the broader regenerative medicine industry, it seems like a lot of commercial stem cell clinics have aggressively tried to categorize their products, including exosomes, under this minimal manipulation 361 umbrella to avoid the whole FDA approval process.
Oh, they have, absolutely. And the FDA has systematically cracked down on it. Exovera Sciences outlines very clearly that exosome preparations universally fail the criteria to be classified as a 361 tissue.
Why is that?
The FDA applies a specific rule, 21 CFR 1271.10, which contains 4 cumulative criteria a product must meet. Exosomes fail almost immediately. First, to acquire exosomes, the source stem cells must be cultured, and the vesicles must be chemically or mechanically isolated, purified, and concentrated.
Which is a lot of work.
Right, that constitutes heavy manufacturing, completely disqualifying them from being minimally manipulated.
And even more fundamentally, the 361 definition specifically applies to articles containing human cells or tissues. But we established earlier that the entire clinical advantage of exosomes is that they are acellular, they don't have cells.
Precisely. Because they are highly processed, acellular fractions of what cells secrete, they're automatically bumped into the much stricter regulatory category, Section 351.
And what does 351 entail?
A 351 biologic is regulated identically to a pharmaceutical drug. That means it requires rigorous pre-market review, multi-phase human clinical trials, and an approved biologics license application, a BLA, before it can be legally marketed to treat any disease or condition.
Okay, but let me point out a glaring contradiction in the current market here, because I can go online right now and find dozens of clinics advertising exosome therapies. They are charging thousands of dollars and claiming to treat joint inflammation, hair loss, and even offering ovarian rejuvenation. How is that functionally happening if the FDA categorizes them as unapproved biologic drugs?
It is happening through a massive exploitation of regulatory gray areas, honestly. And the FDA has issued aggressive public safety notifications specifically addressing it. They have stated unequivocally that there are currently no FDA-approved exosome products for the treatment of any disease.
Zero.
Zero. Any clinic administering an exosome IV and claiming it doesn't fall under biologic drug regulations is operating outside the legal framework.
But the suppliers themselves, companies like Exovera, they're operating legally. The documentation mentions a specific scoping of the FDA's language regarding intended use.
Right. The legal distinction hinges entirely on intent and labeling. The FDA states that exosomes, quote, used to treat diseases and conditions in humans, are regulated as drugs. Therefore, a biomedical supplier can legally isolate, purify, and sell these biomolecular vesicles if they are labeled strictly for topical aesthetic treatments or for medical and biochemical research.
Ah, okay. The product itself is legal to produce and sell under those specific parameters.
Exactly.
But the moment a clinician takes that research grade or topical vial, injects it into a patient's ovary, and claims it will cure their premature ovarian insufficiency, they have crossed the Rubicon. They are making a therapeutic disease claim with an unapproved biologic.
That is the exact line. A supplier can provide a pristine certificate of analysis proving their exosomes are pure, properly sized, and free of endotoxins.
Right.
But purity does not equal regulatory approval. No amount of internal quality control converts an experimental biologic into a legally approved therapeutic drug. If a doctor wants to legally inject exosomes to treat a disease like POI, they absolutely must operate under an investigational new drug application, an IND, within a sanctioned clinical trial.
And the incredible news in this stack of research is that those sanctioned trials are actually underway right now.
They are.
Yeah, the sources pull data directly from clinicaltrials.gov, highlighting 2 active recruiting human studies. For anyone keeping track, that's NCT 06202547 and NCT 06841328. These aren't just abstract concepts in a petri dish anymore. They are actively recruiting young women suffering from idiopathic and medically induced premature ovarian failure.
Which is just so hopeful.
It really is. They are moving forward with testing the safety and efficacy of direct intraovarian injections of mesenchymal stem cell extracellular vesicles.
This is the critical inflection point for the science. We have the foundational animal data, we have mapped the molecular pathways, we understand how miR-21,5P suppresses PTN, and how the hippo pathway is modulated.
We know the mechanics.
Right, we know how the machine works. Now we just have to endure the rigorous, necessary process of proving it translates safely to human physiology over the long term. These human trials are the vital bridge between a miraculous laboratory discovery and a standardized FDA-approved treatment that an oncologist can actually prescribe.
So, what does this all mean for you, the listener tracking the science? When we step back and view this entire landscape, from the cellular destruction of chemotherapy to the nanometer precision of exosomes, we are witnessing a profound paradigm shift in reproductive medicine.
A total rewrite of the rules.
Exactly. Because for decades, the standard of care for a young cancer survivor dealing with POI was purely palliative. You were handed hormone patches, told your biological clock had prematurely shattered, and you were expected to just manage a systemic decline. But exosome therapy represents a complete departure from simply painting over the rust.
It is the transition from symptom management to actual molecular regeneration. We are finally learning how to harness the intrinsic paracrine signaling that our own bodies use to heal, refining it, and deploying it directly into degraded tissues. It is the very real promise of restoring a woman's intrinsic hormonal axis and her fertility, rather than just masking the loss.
And I want to leave you with a final provocative thought that builds on the mechanisms we've explored today. We are currently looking at clinical trials trying to prove that we can use targeted exosome therapy to trick a chemically destroyed, rapidly aged ovary into rebuilding itself and behaving like young tissue again.
Nice.
Well, if that proves successful, the next logical leap is staggering. Could we eventually use this exact same epigenetic reprogramming to delay natural menopause indefinitely?
Oh, wow.
Right. Could the systemic decline of the female reproductive system, what researchers refer to as inflammaging, transition from an inevitable biological fate to a totally optional process in our lifetimes? It's something to think about. Keep a close watch on these clinical trials because the frontier of regenerative medicine is moving faster than we ever imagined.